Good afternoon, everybody. Welcome to today's lunch hour lecture. My name is Stephen Needle. I'm from the UCL School of Pharmacy over in Brunswick Square. I guess it's a truism that antibiotics have changed the world. They've certainly changed everybody's life expectancy in the Western world. However, as I'm sure everybody knows, antibiotics come with increasing problems and challenges. It's with very great pleasure that I introduce today's speaker, Peter Taylor from the School of Pharmacy, who's going to tell us about antibiotics' ever-diminishing returns. Peter is not only an expert, a world-class expert on antibiotics and the mode of action of antibiotics, but he's very fortunate to have, and we're fortunate as well, to have him here with a background that is not purely from academia, but also from many years working in industry, working with the development of new antibiotics. So it's with great pleasure that I ask Peter to present his talk.
Thanks very much, Stephen. I have to apologise from the outset. I've got a stinking cold, so I might have to stop and have a drink of water from time to time. But it's not a bacterial infection, it's a viral infection, so I won't be taking any antibiotics. As Stephen said, antibiotics are incredibly important drugs. When they were introduced in the late 30s, early 1940s, they really did revolutionise the practice of medicine. And they've also had a very big societal impact. But we've been very lax in managing the use of these drugs, and as most of you know, I'm sure you all know, that we now have a big problem with resistance. And there is a view that we might be going back to a pre-antibiotic era when things were really bad. And I don't actually subscribe to that very pessimistic view, but I think we have to be very careful in the future with respect to how we manage these wonderful drugs. So I'm going to, the first part of the talk, I'm going to tell you how we got to this impasse. And for the second part, I'm going to describe quite briefly some of the work ongoing in my laboratory to try and develop slightly new ways of treating bacterial infections that may be able in the future to reduce the emergence of antibiotic resistance. So I'm going to start at the beginning, and I really do mean the beginning. We emerged in Africa about 200,000 years ago. We're the sole surviving species of our genus, which is quite a sobering thought. And for this last 200,000 years, we've had to confront and cope with agents of infectious disease. We've only had antibiotics for about 70 years, so a very, very short period of time. And there have been various challenges that we've had to face up to on this 200,000-year story. And probably the most important thing that happened to us was about 10,000 years ago, some of our ancestors decided to abandon the hunter-gatherer lifestyle, where contact with animals was short and probably very, very violent, to an agrarian lifestyle where we settled down in communities, we bred livestock, and we became much closer to animals and also to the bacteria that populated these animals. And it's no surprise then that most of the infections that we face today are zoonoses. These are infections that man has inherited from animals. And this really has driven the panoply of infectious disease agents that we have to confront. And I believe that infectious diseases have actually shaped our history. And just to give you a few examples of how antibiotics have had a big impact on society, the only time the population of Europe went down was in the 13th, 14th, and 15th centuries, when it was halved due to waves of epidemics, due to smallpox, which is a virus, but for typhus and particularly plague, which are bacterial infections, and they were really devastating. And we had really very little around to combat these pandemics, these epidemics, and in fact we didn't even know they were due to infectious agents. A bit nearer our time, it was in the 1930s even that many birthing mothers would acquire streptococcal infections, which were life-threatening infections, and it was only due to the introduction of penicillin in the early 1940s that these infections, known as puerperal sepsis, became almost a thing of the past. You probably, if you read the history books, you probably read that Napoleon Bonaparte's army perished at the gates of Moscow because of the severe Russian winter. That isn't strictly true. They were bitten by lice. They acquired typhus, which is a bacterial infection, and the number of troops was decimated here. And worse than that, the troops staggered home, and they brought with them the typhus bacterium, and they infected many, many people in Western Europe, and about 2 million were killed. Only 100 years ago, the 3 biggest killers in the United States were all bacterial infections: tuberculosis, pneumonia, and bacterial gastrointestinal infections. Only one of these now is in the top 10 list of reasons for mortality in the States. And in case you're wondering, that's pneumonia. Upper respiratory infections and lower respiratory infections never really seem to go away. So all this changed about 70 years ago when the first antibiotics were introduced. And they really did have a huge impact on the practice of medicine. And here's some images from one of the very first patients to receive penicillin G by injection. This was a young girl who was very close to death, started getting injections of what was in those days a very precious resource, and within 3 or 4 days you can see that she started to recover, and within a few weeks she was fit and healthy again. This would not have happened without antibiotics. This girl would have died. And this so-called golden age of antibiotics started in 1941 when penicillin G was first introduced into the clinic. And this launched a very successful search for a range of antibiotics, and I've listed some of them here, and most of these are in use today. So we have to thank scientists in the 50s and 60s for providing us with these wonderful molecules. So cephalosporins belong to the same class as penicillins. They're beta-lactams, and drugs like vancomycin is a lifesaver if you have an MRSA infection, and gentamicin still proves very useful as an agent for treating severe infections. With sanitation measures, antibiotics have had an enormous impact on life expectancy. Early last century, life expectancy of an average male was 47 years. In 1950, it was early 60s. Now it's 79. And a lot of this is due to antibiotics and to public health sanitation measures, immunization that prevent us acquiring microbial infections. But it goes further than that. The reason we can undertake organ transplantation, while we can safely care for babies who are preterm, why we can indulge in cancer chemotherapy, and we can have many intensive care interventions, is simply due to the fact we can provide antibiotic cover against a wide range of bacterial infections that will threaten these patients who are probably severely immunocompromised. So these really are miracle drugs. These are wonderful molecules, and we should have preserved them a lot better than we have. Unfortunately, we didn't do that. We were very lax in our management of these molecules, and we now face a situation where we have a very large number of infections worldwide due to antibiotic-resistant organisms. Some people saw this coming. Alexander Fleming discovered penicillin just a few miles from here in St. Mary's Hospital, and he got the Nobel Prize in 1945. And during his Nobel address, he came up with something very prescient. Remember, this is 1945. He said you can expose bacteria to suboptimal doses of antibiotics in the lab, and you can engender resistance. And he said that there is a danger that the ignorant man may easily underdose himself, and by exposing his microbes to non-lethal quantities of the drug, make them resistant. This is exactly what's happened. We haven't managed these drugs very well, so we have lots of resistance due to suboptimal dosing of these molecules. There were other warning signs. In the early 1950s, a multidrug-resistant strain of the dysentery bacillus emerged in Japan, and people thought this was a one-off. This organism was resistant to 6 different antibiotics and was getting close to being untreatable. And also during that time, we started to encounter drug-resistant tuberculosis. Excuse me. And tuberculosis is carried by 1/3 of the world's population. There are 8 million new cases every year, and there are 1.7 million deaths every year. So this is a very important infection. And the last thing we want to do is face organisms which are resistant to the frontline drugs that we want to use. So in spite of all this, people were very complacent, and Dr. William Stewart was the senior medical officer in the United States. I don't know whether he's still alive, but if he is, I'm sure he wishes he'd never said this. He told Congress in 1969 that we conquered infectious diseases. The time has come to close the book on infectious disease. Nothing can be further from the truth. This in some ways acted as a trigger for major drug companies to begin to wind down their drug discovery programs in antibiotic chemotherapy. And this wasn't a very good thing. Why is this? What's special about antibiotics? Well, if you take a normal drug, it interacts with the patient. It might have some toxicity. Hopefully it will have a successful clinical effect. And everything stops there. So if you were to develop, say, a beta blocker for the treatment of cardiac arrhythmias or hypertension in 2013, it will be just as good in 100 years' time as it is now. It will be superseded by better drugs, but it would still work just as effectively. That isn't true of antibiotics. Antibiotics have an effect on the patient, obviously. They're relatively non-toxic, so toxicity isn't a big problem with antibiotics. They may have a clinical effect, cure the patient, but if the dosing is suboptimal, they'll engender resistance in the bacteria that's causing the infection. And the best way to engender resistance is to treat the bacteria with suboptimal amounts of antibiotic. And if that happens, there are all sorts of ramifications. There'll be treatment failure. The resistance can be transmitted from one bacterium to another, and the resistant bacteria can be transmitted from one patient to another. These resistant bacteria often colonize the patient, and they're excreted into the environment. If you go to India, where they produce lots of generic antibiotics, and sample the water downstream from plants that manufacture antibiotics, virtually all the bacteria will be multidrug-resistant. They get into the environment just because we excrete the bacteria, the antibiotic in our urine or our feces. Doesn't mean to say that's the end of the story. They'll contaminate the environment and drive resistance from within the environment. And the most important thing is to stop this happening by not using antibiotics inappropriately. Well, you might ask, why don't we just make some new antibiotics? Virtually all the antibiotics, with the exception of Penicillin G, which came from laboratories in Oxford and from companies in the States. Virtually all the antibiotics we have been developed within the pharmaceutical industry. But the pharmaceutical industry aren't making many antibiotics anymore. And this graph here shows the number of new introductions of antibiotics in the United States over 5-year periods beginning in 1983. And you can see this was at the end of the golden age of antibiotic development. There were 16 new drugs introduced onto the market for the treatment of bacterial infections. This has declined for every 5-year period, and now this has been completed. So for 2008 to 12, there have just been 2 new introductions, and these aren't fundamentally new drugs. They're so-called me-toos based on existing known structures. And you don't have to be a mathematical genius to work out that if this continues for the next 5-year period, there won't be any new drugs available, and yet resistance moves on unabated. The reasons for this are complicated. There are fewer major drug companies now. There were 30 25 years ago. There are now 6. They've grown big because of mergers and acquisitions. And antibiotics are not particularly attractive candidates for drug discovery in the pharmaceutical industry. They're at the fringe of economic viability. You have to invest $1,000 million to develop one new drug these days, and the industry are concerned they're not going to get their money back. So what they do now is they develop so-called lifestyle products. They're very interested in anti-obesity drugs, and there are more anti-obesity drugs in clinical development today than there are antibiotics, which I think says it all, really. For the industry, they're unattractive. We use them to treat acute diseases, so short courses, 1 or 2 weeks. Contrast that with an anti-obesity drug that will probably be taken by the individual, because they're not patients, it's a lifestyle issue. They'll take it for the rest of their lives. Antibiotics are very cheap. They're cheap and cheerful drugs. People expect them to be cheap. And this has obviously pushed down the pharmaceutical industry's interest in this whole field. There's also a curtailment of unnecessary use, which means if we develop a fundamentally new, good drug, we want to keep it back for those really problematical infections. So this is no good for the industry. They're not going to be using these drugs as often as they would like. I think one of the biggest issues, though, is that the industry forgot how to make antibiotics. Virtually all antibiotics come from fermentation broths produced by bacteria from the soil. And all the wonderful drugs we have, they're very complex chemically. No chemist would ever make them. But in the 1980s, the industry decided it was going to enter the new field of molecular biology and have high-throughput chemical screens against biochemical drug targets. And they spent $17 billion doing this, and they came up with precisely no drugs at all. So they've lost interest. So why does resistance emerge so quickly? Well, it's probably quite appropriate that I'm giving this lecture in the Darwin lecture theatre, because really the emergence of antibiotic resistance is an example of Darwinian natural selection, but it's an extreme example. The modern horse took 50 million years to evolve from the Eohippus precursor. MRSA took 3 months to appear due to the selective pressure of antibiotics. So if you're looking for a good Christmas present for anybody in your family, I'd highly recommend this book, The Origin of the Species, by Charles Darwin. The most wonderful read, written at a time when people thought the world began on the 23rd of October, 4004 BC. So it's a monumental work, and what we see with antibiotics is extreme selective pressure on bacterial populations. There'll always be 1 or 2 pre-existing resistant strains or mutants within a bacterial population, and if we're not careful with our antibiotic, we will kill all the susceptible bugs, and we will be left with the resistant organisms which can go on and cause more problematical infections. And the problem is, the pressure for this to occur is the presence of antibiotics, and they're everywhere. They're in the patient, of course, but there are many antibiotic residues in our hospitals which drive resistance, and also, as I mentioned before, in the environment. So what sort of bacteria are we talking about here? They come and go, and this is part of the problem. 15 years ago, everybody was worried about MRSA. That's Methicillin-resistant Staphylococcus aureus. This is still a big problem, but not such a great problem as it was, maybe 10 years ago. The big problem today, in the UK and in Western Europe at least, are infections due to so-called gram-negative bacteria, and you can see Escherichia coli, which was considered a few years ago to be a relatively benign organism that is a natural commensal in our guts, causes 36% of all blood infections in England and Wales in the last year of survey. And this is the big problem. And these are multidrug-resistant bacteria, and they're very, very hard to treat. They're verging on the untreatable. And in some hospitals, the chances of survival with an infection with one of these bacteria, a bloodstream infection, is less than 20%. So these are really nasty bacteria. And there are others. TB will always be a big problem. And Clostridium difficile, C. diff, I'm sure you've read about that in the newspapers, is an infection that's causing lots of problems in our hospitals, although it does seem to be slightly on the wane. It's a global problem. Resistance is not a problem restricted to any one country. These bacteria, they don't have passports, but they don't recognize boundaries. With modern communications, they can be spread around the globe very, very quickly. And we use antibiotics in the food chain. High-density farming requires antibiotics as growth promoters. If an animal in a herd gets sick, we don't just treat the sick animal, we treat the whole herd. More than half of the antibiotics that we use are used for animal husbandry, and this means that they get into the food chain. The antibiotic residues and the bacteria will get into the food chain. It's pretty clear that if you have an infection due to a multidrug-resistant bacterium, it increases the chance of death and serious morbidity. Also, there's a huge burden on health services. In the States, it's been recently calculated that the cost of an infection due to a multidrug-resistant organism increases treatment costs by $18,000 to $29,000 of dollars for each patient. So this has an economic impact as well as a human impact. And just to emphasize this, let's follow the lifestyle of one of our superbugs, MRSA, antibiotic-resistant Staphylococcus aureus. When penicillin was introduced in 1941, virtually all staphylococci were very, very susceptible to penicillin G. But that didn't last very long. The bacteria started to produce enzymes which broke down these drugs. But that was okay because the industry got very busy. The industry got very busy and made lots of wonderful derivatives of penicillin, and some of them are here: oxacillin, flucloxacillin, and methicillin, after which MRSA takes its name. And these were developed because these antibiotics were resistant to the enzymes that the bacteria produce to break them down. So methicillin was the first of these to be introduced in 1960, and 3 months later, the first MRSA strain appeared in a hospital in Kettering in the UK. So Kettering is famous for this reason. It gave rise to the very first strain of MRSA. And MRSA incidence has increased year on year since 1960, but that's okay because we can use another antibiotic, gentamicin, to treat these patients who are no longer susceptible to a therapy with methicillin. But then gentamicin resistance appeared, and so this bug is slowly accumulating resistances to many, many antibiotics. But never mind, because so-called glycopeptide antibiotics were introduced in the early 1980s, vancomycin and teicoplanin, and these remain the mainstay of our arsenal to treat these multidrug-resistant infections. Unfortunately, in 1996, vancomycin intermediate resistant strains started to appear. And these appeared, first of all, in Japan, where they used more antibiotics than anybody else. And these strains were only a little bit more resistant to vancomycin than the standard strains. But that difference meant the difference between clinical success and clinical failure. So this is sort of contained, but we get these strains in the UK now. And in 2002, the nightmare scenario came to pass, where full-blooded vancomycin resistance emerged. And these organisms, vancomycin-resistant Staphylococcus aureus, have been so far kept in control. There have been 5 outbreaks in the United States, but sooner or later, these are going to get out into the wider hospital population. We have 2 new drugs that were introduced into the market about 13 years ago, linezolid and daptomycin. These were introduced specifically to treat MRSA infections, and resistance to these drugs is already emerging. So if we get VRSA combined with resistance to these drugs, staphylococci will give rise to untreatable infections, which isn't a very nice thought. So what we try and do in my lab is to look at different ways to treat these infections. How can we break this cycle of a new antibiotic, introducing it into the clinic, and then resistance emerging in a very short period of time? Well, I think there are alternatives to conventional antimicrobial chemotherapy, and I think people are beginning now to listen to what people like I have got to say. And I take my cues from this wonderful individual, one of my heroes of the 20th century. René Dubos was a microbiologist at the Rockefeller University in New York, and he said bacteria are only opportunistic invaders of tissues already weakened by crumbling defenses. And what he meant by that is we normally have a fine balance between the microbes in our body and resistance to infection. But sometimes this balance can be tilted in favor of the microbe because we're weakening of our immune system, and then you have infection. So our approach is to try and restore the balance. Instead of killing the bacteria, why not try and restore this balance to give the host a chance of clearing the infection? So we know that introduction of a new antibiotic always leads to selection of drug-resistant forms. So we're not trying to kill the bacterium, we're trying to disable it to reduce its chances of surviving in the host. And if you think about that for a minute, if we use this strategy, we're not applying so much Darwinian selective pressure on the bacterial population, because our modifying agent isn't actually killing the bacteria, it's just making them less fit. So that's an unproven hypothesis, but it is our working hypothesis. And we call this modifying the bacterial phenotype to make them less fit. And there are 2 ways we can do this. We can look at multidrug-resistant bacteria and say, can we modify the bacterium so that they become susceptible to conventional antibiotics that have lost their utility because of the emergence of resistance? So what you would like here is an agent which modifies resistance to make the bacteria more susceptible to conventional drugs, which you can then use, and hopefully you'll get treatment success. And the other way of looking at this is bacteria that cause infections normally have an arsenal of virulence factors which enable them to cause infection, to damage tissue, and to spread and survive in the body. What if you can compromise the expression of those factors? You'll have attenuated bacteria, less fit bacteria, that then could be cleared by the immune system. So we're trying to compromise resistance to drugs and compromise the capacity of the bacteria to cause serious infectious disease. So to take a look at the first example, we looked to see whether we could increase the susceptibility of MRSA strains to cheap drugs that are now ineffective due to the emergence of resistance. And to do this, we've looked to the Far East, to Japan, where studies were done in the early 1970s in Shizuoka Prefecture underneath Mount Fuji. These people there, they don't live forever, but they've got the highest longevity of any substantial population in the world. And they drink many cups of green tea every day. People started to look to see what was in green tea. And there are a number of quite complex chemicals called catechins. They're in the polyphenol group. And there are two which are very abundant. This is epicatechin gallate and epigallocatechin gallate. And these molecules have a number of very interesting properties. Epigallocatechin gallate is a very good antioxidant, and it's actually in clinical trials at the moment as a supplement to the treatment of various cancers. And there's a lot of information about this molecule. When we're interested in epicatechin gallate, which is this chemical structure here, the gallate group is this fused ring system here, which is very important for activity. And although this compound only has very weak antibacterial properties, it can protect against dental caries. There have been some clinical studies undertaken to show that if you drink lots of green tea or buy tablets from Mitsui Norin in Japan and use these as oral washes, you can prevent dental caries. But they have some very interesting interactions with bacteria. And this is what we found when we added ECG to conventionally growing MRSA strains. This is what the control looks like, no ECG. These are Staphylococcus cells, they're round cells, they're cocci, and they're dividing quite nicely through binary fission. One cell separates into two daughter cells ad infinitum until they run out of food. But when we grow them in the presence of ECG, they grow just as quickly, but they change their shape rather profoundly. The cells become bigger, they don't separate very well, and the cell walls are very thick. And this is manifested in the fact that equal weights of bacteria grown in the absence or the presence of ECG occupy different volumes. So this renders the bacteria much less dense, and they have these very thick, less dense cell walls. But they survive quite happily, and these are scanning electron micrographs of the control cells and the ECG-treated cells, and you can see they're bigger, they're not separating, and they have a very rough surface. But one of the really astounding things about ECG is it has a huge capacity to modify the drug resistance of MRSA. So beta-lactams, to remind you of the drugs of the penicillin and cephalosporin classes, and MRSA is usually resistant to very, very high concentrations of oxacillin. So 512 milligrams per litre, these are not doses you'd ever be able to replicate in the body. This drug is resistant to huge amounts of oxacillin, which is a conventional second-generation beta-lactam drug. This means that you need 512 milligrams a litre to prevent overnight growth of this organism. Huge amounts of drug. If you grow these organisms in the presence of the non-gallate catechins, they don't have this gallate group here, nothing much happens. But if you use the catechins that have the gallate group, you can see that they start to become more susceptible to oxacillin and also to all other beta-lactam drugs that we've measured. And ECG gives this remarkable effect. The minimum inhibitory concentration, the amount of drug necessary to prevent overnight growth, goes from 512 all the way down to 1. And it goes below what's called the antibiotic breakpoint, which means it becomes clinically very, very interesting indeed. So these compounds don't have any effect on the viability of these bacteria, but they can render MRSA bacteria totally susceptible to beta-lactam drugs. So How's this happened? Well, this slide actually encompasses about 10 years' work in my laboratory. MRSA is resistant because it has an extra protein which sits alongside this protein here, which is called penicillin binding protein 2. Penicillins inactivate this protein. It no longer functions, but if this molecule is in close proximity, it will take over some of the functions of PBP2 that have been inhibited by the antibiotic, and these cells will continue to grow in the presence of high concentrations of beta-lactam antibiotics. But what ECG does, it inserts into the membrane of these bacteria, and it dissociates PBP2 from PBP2A. And because this happens, these two can no longer cooperate, and the cells become completely susceptible to oxacillin and other beta-lactam antibiotics. So I'll quickly move on to the other paradigm, which is to try and help the host immune system to remove dangerous bacteria by disarming the pathogen. And one of the diseases we're very interested in is sepsis in the newborn infant. This is a major killer of neonates, and the newborn infants acquire bacteria after penetration of certain bacteria through the skin or translocation of bacteria from cells within the gut, mucosal surface generally in the gut. They invade the circulation, they cause a nasty inflammatory response, and they can sometimes target specific organs such as the brain, where it causes meningitis, and this is a disease that's quite common in the UK. 1 in 3,000 newborns will get bacterial meningitis. The outcome is not good, even with the best treatments. Only 90% survive, and if these infections go untreated, they're usually fatal. One of the really interesting things is that all bacteria that cause meningitis, and E. coli is a very good example, carry a protective capsule around the cell. So what we want to try and do is somehow remove this capsule at the site of infection to sensitize this bacterium to the host immune system. The host immune system will be able to clear these organisms relatively readily. So where did we get such an agent from? Well, bacteriophages are growing in popularity as means of treating infections from cells, and these are viruses that cannibalize bacteria. They bind to the surface, inject their DNA, take over the cell's replicative machinery, and you get lots of new particles generated. These phages have a problem getting through encapsulated organisms. They need to digest their way through the capsule, so they carry enzymes as part of their tail base, the spikes here. And we've basically taken these enzymes and we've tried to develop them as therapeutic agents. Now, this is what phages generally look like. If they carry a capsule degrading enzyme, not all of it's incorporated into the bacteriophage, so you see this halo effect. This is just free enzyme diffusing out from the plaque. And this is, we've taken advantage of this to clone and engineer this protein to enable to use it as a therapeutic. Now, E. coli K1, which is the main cause of E. coli neonatal meningitis, colonizes the gut. It then traffics through the circulation to the brain, and it needs a capsule to do that. And these are smears from blood, and this green stain is the capsule. It's very well expressed during transit from the gut into the brain. We have a rodent model of neonatal infection, and in the untreated animals, we can colonize the gut very, very readily, and the bacteria get a blood-borne infection, and if we allow them to continue to turn, which we don't do, they will all die. If we give one shot, very, very small amounts of our enzyme, very early on in this process, we can prevent both bacteremia and death from this potentially lethal infection. And we can give it later on down here. The longer we leave it, the less effective it is. But that seems to be a really good way to handle these organisms. But E. coli K1 is just one of a very limited number of organisms which can cause meningitis. So meningitis isn't the best target for these for this enzyme approach. But we think that anthrax is. Anthrax is a very rare disease in the Western world. But the interesting thing about it is there's only one pathogen associated with anthrax, and that's Bacillus anthracis. All strains produce a unique capsule. The capsule is absolutely essential for the capacity of this organism to cause infection, and we need to protect ourselves against B. anthracis because it's a very attractive weapon for people who wish us harm. And up to 10 drug resistance elements have been engineered into the pathogen against all international conventions. So we really need to do something about this. And so we've used this enzyme approach. We couldn't find any phages that produce the enzyme. So my colleague David Negus in my lab went to the soil outside the School of Pharmacy in Brunswick Square and used media containing minimal salts and only the capsular material from B. anthracis as a sole source of carbon and energy. The principle being that if any bacteria in the soil can degrade this molecule, they're going to have an enzyme that breaks it down into small pieces. And that's what we found. We've isolated an enzyme and we're developing it now. This shows you what it can do. We call our enzyme ENVD. This is the Anthrax bacillus. 5 minutes after treatment with the enzyme, the capsule is completely gone. So we're in the process now of developing this with our colleagues at Porton Down, and we think this shows quite a great deal of promise. Things aren't as bad as we think they are. We've got lots of articles like this. We failed to protect a precious resource, but there is a high level of concern now. Very much of it is due to our Chief Medical Officer, Sally Davis, who's really driven this issue up to the top of the agenda. She's written books and ensured that very comprehensive reports have been made, and there are other initiatives in Europe and in the UK that have brought this issue to the public's attention. Even drug companies are getting a bit interested in this topic now. So things aren't as bad as you think they might be. So all that remains for me to do is to thank the people in my lab who do the hard work. They're in the audience here, and to thank all these various organisations for providing us with the money to do this work. Thanks very much.
Thanks very much, Peter, for a tremendous talk, a real insight into where we might be going. It's, of course, ironic, but positively ironic, that you're now finding leads from the soil, which is where so many antibiotics came so many years ago.
Yeah, absolutely. Yeah. There was a fundamentally new class of antibiotic was introduced, was published about 6 months ago, and it came from a bacterium in the sea. It was an actinomycete, and most of our antibiotics come from actinomycetes, and there are 10 to the 29th bacteria in the oceans of the world, and some of them must be making compounds that could be developed into drugs. And I think we've spent all our time looking in the soil. Maybe we should consider other complex environments, such as the oceans of the world, to try and find fundamentally new sources for antibiotics.
Indeed. Well, we've got time for a few questions. One over there, I think we've got also roving mics, so please use the mics.
Hi, good afternoon. Very good talk. Nick Dimitris, ex-UCL. 2 questions. You kind of alluded to this in the beginning, but who would you take antibiotics? Sorry. If you took antibiotics away from the pharmaceutical industry, who would take control? That's the first question. Second, resistance to new penicillin or non-penicillin-based antibiotics is a public health issue. What about population shifts and lack of disclosure from company to company because of patent issues, etc.?
Yeah, the patent issue is an important one because it takes on average 11 years to develop a new drug, and you get patent protection for 20. The regulatory authorities are trying to address this issue. And also, with most drugs, I mean, until very recently, the FDA said for a new drug to be introduced, it must be more effective and less toxic than existing molecules. And that isn't the issue with antibiotics. Antibiotics are incredibly non-toxic. You can take 10 grams of penicillin, it won't do you any harm. The problem, and there's no drug more efficacious than penicillin, it just works wonders. The problem is resistance, and it's only recently that the regulatory authorities have woken up to that. It's also very difficult to do clinical trials in this area because you can't simply take a patient population, divide it into 2 and say you'll give one half your experimental drug and the other half a conventional drug and do a comparison. People, you can't predict when infectious diseases with different organisms are going to occur. It's very, very difficult to do that. So development is fraught. So I didn't get your first question.
The first one was about who would you, if you took antibiotics away from the pharmaceutical industry, who would take charge?
That's a very good question. I mean, the industry are getting interested again, because if you think about it, they're not going to be able to sell other drugs if we can't perform major surgery. It's going to have a huge impact on health. But I don't think it's fair to expect the pharmaceutical industry to develop the sort of molecules they did in the past. And there are moves afoot now for really a three-way collaboration between governments. Governments must take a lead here. Between governments, the pharmaceutical industry, but I think also academics. We need to do something. We need to drive new paradigms of treatment, because the industry is relatively risk adverse. It's not going to do this by themselves. And I really do believe that this is, what we do is just a small number of different things. There are other people out there doing other unconventional things that might, at the end of the day, give rise to new antibiotics. So I think it should be a three-way collaboration.
Can we actually move on to somebody else who might have a question? We've only got a very limited time. Last question, I'm afraid. We have to be out of here by before 2 o'clock.
Hello.
We could go on forever, I'm sure.
Thanks for the great talk. I wanted to ask you, in a public health perspective, maybe it's somewhat unrelated. There seems to be developing in population a new misconception about medicine. I'm thinking about the stance about vaccines, for example, in some parts of the US. Do you think that on the antibiotic side, this could, the fact that people seem not to trust these things anymore for some reason, will it create problems in your opinion?
Yeah, I don't see mistrust on behalf of the patient population. If you think back to those images of that young girl who was dying and got an antibiotic and her life was saved, why wouldn't you want to take an antibiotic? I know there are sort of homeopathic movements around that would rather we just ate jungle juice, but at the end of the day, serious life-threatening infections are going to have devastating effects. And if we can get antibiotics that work, if we could serve what we have, then I don't see any objections to people taking antibiotics.
There's people who might outside there, and I think it's an increasing trend that people, in many places, they just say, I'm not going to take antibiotics because they are bad for me. And I don't think that this is true, but it seems to me that there is this trend of people getting more worried about this.
Well, in the past, antibiotics have been prescribed for conditions where they wouldn't have any effect. For example, a few years ago, people went to their GP with a chest infection like I've got due to a virus, and they expected to receive antibiotics, which would do no good at all. I think now we've handled that issue. I think now the general public is much more aware that they shouldn't ask for antibiotics unless a bacterial infection has been diagnosed. So I think we do need to conserve what we've got in that manner. But of course, it's a free world. If it's your choice not to take an antibiotic, then that's your choice.
Right. I think we have to stop here. Let's thank Peter very much indeed for tremendous talk.